Okay, this set of notes is on membrane structure and function. This is chapter 7 of your textbook. This will include the cell membrane information. Hold on. This will include the cell membrane as well as cell transport. If you remember that from biology, that's lovely osmosis and diffusion information. We are not covering chapter 6 because you did that in your presentations. Those are all of the cell parts that you need to know. The cell membrane is also called the plasma membrane. So if you hear the term plasma membrane, it's talking about the same thing. You might also hear it called a phospholipid bilayer. Those are some different terms you could hear, and they all mean the cell membrane. The cell membrane is a fluid mosaic model. The reason they have is a fluid mosaic model is because it moves fluidly. It's not a static structure. It is not going to remain in place. It actually moves, and I have a couple of videos for you here in just a second on that. There have been many proposed models over time of the cell membrane, and this is the most current, is the fluid mosaic model, because we've learned more and more over time about how that cell membrane is structurally put together and how it works to do its job. And we're not concerned too much about the history of models that have been proposed in the past. However, your textbook does have some information about that if you're interested in that history. Lipids and proteins are the main components of the cell membrane. Phospholipids are the main lipids. You've learned about phospholipids in Biology 1. Whether you remember them or not is another question entirely. A phospholipid has a hydrophilic portion and a hydrophobic portion. The hydrophilic portion is attracted to water, and the hydrophobic portion fears water. It is the part that is away from the water. That's the tail. I always think of this as a remembrance by a dog. When a dog is afraid, they hide their tail. They tuck their tail. Well, in the cell membrane, the phobic region tucks its tails. We call this amphipathic. It has two regions. So here's a picture of what that looks like. These heads are water-loving, and the tails are water-fearing. This is why we have water on the inside of the cell and water on the outside of the cell. And the tails are right here in the middle where they are not exposed to the inside of the cell or the outside of the cell. This is a molecular picture of a phospholipid. This is the head, this is the tail. The tail is a lipid. We've shown you, you've probably seen this before, when we did one of the chapters, there was chapter, I think, 5 on the large biological molecules. This is a picture of a cell membrane, or a cutout of a cell membrane. The cell membrane is mainly made up of these phospholipids. It's a bilayer because there's one layer here and one layer here. All right, sorry about that. It's kind of hard to work at home sometimes. And then these are proteins that are embedded in that membrane, and that is for transport. It allows things to go in and out. Some of these proteins actually span the entire membrane, and then there'll be some that actually sit on the surface of the membrane that don't actually span the whole thing. So here's a really complicated picture of the cell membrane. These are fibers that hold it kind of steady, or inside the cell, kind of, these are the extracellular matrix. So outside the cell, just fibers that you're going to find all over. And then you've got the cytoskeleton, which are fibers inside the cell that hold the structure of that cell. These are those proteins we talked about. There are also carbohydrates that are attached to some of the proteins that extend off of it. There are things called glycolipids, which are not carbohydrates, but they're lipids that extend off of those proteins. There are, integral proteins, which span the entire membrane. Then there are peripheral proteins which lay on the outer edges. I talked about that a little bit before. And then there's cholesterol, which we'll talk about its purpose a little bit later. But there's cholesterol also embedded in that membrane. Lateral movement of phospholipids within the membrane is rapid, but the basic structure remains because of its polarity. Remember, these heads up here are hydrophilic. They're okay with the water. The tails are hydrophobic. They are not okay with the water. And so no matter what, if you puncture this membrane, if you stabbed at it, it might actually come apart, but it would remain back in the same position. Because of the polarity, these parts are going to remain in this orientation. And I'm going to show you some models of that here in a little bit. The membrane remains fluid as temperature decreases, but the phospholipids become more closely packed until it solidifies. Think of baking grease cooling. Some membranes are more fluid than others, and that just depends on species to species. So this is showing you the lateral movement that can actually occur. So these can flip-flop here, or they can actually flop across the membrane and be exchanged. That is perfectly normal as long as when it all is said and done, they go back to the same arrangement. Cholesterol actually makes the membrane less fluid by restraining phospholipid movement, but at the same time, it lowers the temperature at which the membrane can solidify. So cholesterol actually plays kind of a double role here. It makes the membrane less fluid, but also makes it be more fluid. I know that sounds kind of counterproductive there, but it wants to make sure that the fluid is basically not changing the fluidity. It kind of keeps it buffers that change. It causes it from not making too much of a change in the fluidity of it. We also, scientists have found that the lipid composition of cell membranes varies from species to species because of different environmental conditions. And that's just an evolutionary adaptation. The cell membrane may be more fluid or less fluid, depending on the species and how much lipid that they have in there. This is showing some different levels of fluidity. So this is a membrane that's really fluid. It can move a lot. It has lots of these bent tails. Remember when we went back to the macromolecules, we kind of talked about that bent tail with the double bond, and that allows it to not be very tightly packed. Things that are unsaturated fatty acids are going to have more of these bent tails, that double bond, and that causes them to be liquid at room temperature. And then this is a very viscous, I think is how you pronounce that, cell membrane, where the tails are very saturated, and so they don't have that bend. And they can pack really tightly together and cause the membrane to be less fluid. Here's pictures of cholesterol. And cholesterol's job is basically to maintain the fluidity that is normal for that particular cell membrane. Cell membrane proteins. Proteins determine most of the membrane's function. Okay, different types of cells contain different proteins. This also applies to membranes within a cell. So not all cell membranes are different. Maybe the cell membrane of skin cells is going to be different than the cell membrane of nerve cells because they're doing different things. Integral proteins, which I pointed out before, penetrate the entire hydrophobic interior. They span the entire membrane. Peripheral proteins adhere loosely to the surface of the cell membrane, and sometimes to parts of integral proteins. And this is showing a protein. Remember, we show them as these big blobs a lot of the time, but we have to remember that they are proteins, which means they are made out of amino acids, and that they have a primary structure and a secondary structure and a tertiary and oftentimes a quaternary structure. So we're showing either the tertiary or the quaternary structure, just the big blob. But I like this picture because it also indicates to you that there is another structure kind of inside there. That these proteins are not really, they don't really look like big blobs. But that's just easier to draw for the purposes of what you need to understand. What are some of the functions of the membrane proteins? They help to transport. That's the big one we're going to be focusing on in this particular unit. Enzyme activity, they also play some important roles in enzyme activity. Signal transduction, that's more cell communication in Unit 3, and then again in Unit 6. We'll talk more about that, but they can actually be receptors to allow things into the cell. Cell-to-cell recognition, so one cell might recognize another based on the proteins that are on the outside of the membrane. And intercellular joining, so joining cells together. Attachment of the cytoskeleton and extracellular matrix helps provide structure to the cell. Notice I showed you those filaments that were both inside and outside the cell. So this is the inside of the cell. And this is the extracellular. You ever see ECM, that's extracellular matrix. That's what's outside the cell. A cell membrane may have several different proteins, and also a protein may have multiple functions. It may not just do one job. Here are some examples of these. So transport, this is what we're going to be focusing on. Enzyme activity, so causing reactions to happen or not happen. This is what I was talking about with signal transduction. A signaling molecule may be picked up by a receptor on a protein, and then that signal is sent to do something inside the cell so that an action can happen. Cell-to-cell recognition, so this is going to recognize one cell membrane to another, so this skin cell might recognize that skin cell. Intercellular joining, so that cells can join together. Think about how tightly closely packed some cells are to each other. And the attachment of the cytoskeleton and the ECM to those proteins here. You can see that attachment on both ends. Cell membrane carbohydrates. Carbohydrates on the cell membrane aid in cells being able to recognize each other. They're all going to have different carbohydrates. One of the biggest examples of this is the ABO blood type. The reason you would have A, B, or O blood type is based on what carbohydrates you have. You can have A carbohydrate, which might have like a shape like this, or a B carbohydrate, which can have a shape like this, or you might have none. Or you could have both carbohydrates on your on the surface of those cells. Glycolipids are bonded to lipids, and glycoproteins are bonded to the proteins. They vary from species to species, organism to organism, so the ones that you have might not be the same as the ones I have, and even cell to cell. So the skin carbohydrates might not be the same as your blood cell carbohydrates. They're very unique. That's what they're saying. This is showing how the HIV virus works based on those receptor proteins. Remember we said proteins, here's the cell membrane right here, the protein, and the reason that the HIV virus is allowed inside the T cells is because these receptors recognize a receptor on the HIV virus, and that allows it to gain entry into the cell, and then of course this is RNA, genetic material. It can actually reproduce and make more HIV viruses that will then exit the cell and go and infect other cells. And this is showing membrane glycolipids, secreted proteins here that are being secreted. So when we talked about all the cell parts, remember that proteins are made on the ribosomes. Okay, they are packaged into vesicles. Those vesicles fuse on one side of the Golgi, exit on the other side of the Golgi, and then are fused with the cell membrane and then are released onto the outside of the cell. Now, we are going to talk about cell membrane permeability in another video. So I'm going to shut this down real quick. I'll keep it for right now. pull up the web. I had a couple different videos I wanted you to look at. Now, when you see this video, you can go to these videos, so I'm not going to play the whole video. The first one I want you to go is to this one. So the website address is right up here at the top. You could type that in, or you could just search for model of the fluid mosaic model and look for this picture in this video. It's about 5 minutes long. This is a little easy model that this, it sounds like maybe like a TA at a university or something, that he made to demonstrate how the cell membrane acts as a fluid model. Then I want you to watch this one, and then I want you to watch this one, and you can either copy that information down for the web address there, or you can search for it, fluid mosaic model, and this looks like it is on DNAtube, a scientific video site. That's what its website is. Either way, when you play this one, it's only about, looks like 1 minute and 26 seconds. What I want you to do after watching both of these videos is in the notes that you're taking for this video, I want you to write down in this model, what are some positives about his model? What are some things that really help represent the actual structure of the cell membrane? But also, what are some of the downfalls? What are some things that aren't really accounted for about the cell membrane in this video? And then when you look at this one, I want you to think about what good things are about this model, about what the animation is showing you. And then what are some of the downfalls? What is it not also showing you? So good and bad for both these videos. Because I really like this one, but there's some things that's really leaving out. I like this one because it's really giving you a little bit more depth and detail to how it works. However, there's some limitations with this one as well, as well as the fact that you can't build it like you can build this one. So I want you to go and watch those videos and then fill those notes in, write a little bit of information about those videos in your notes for class.